Hi, my name's Leeanne McGurk, and I'm a researcher at the School of Life Sciences at the University of Dundee.
And I'm Melissa Parra, and I am a PhD student in Leanne's lab in University of Dundee.
And our lab is focused on understanding motor neuron disease. And we specifically look at a protein that has the name TDP-43. And we'd like to tell you a little bit about what TDP-43, what this protein does in our brain cells. And to do that, we'd like to introduce you to our demonstration. This really represents any cell in the body. Today we're considering the brain cell, and it's divided into two compartments, the nucleus and the cell body. Now our protein TDP-43, it's this green ball. It mainly locates itself in the nucleus, and its job is to read our instruction manual. So every cell has an instruction manual. It's our DNA, it's our genetic information. It's what tells our cells how to be a cell and what to do. And TDP-43 helps us read this instruction manual. And by reading the instruction manual, the cell knows how to function properly or how to work properly. Now, although it's mainly in the nucleus, it does go out into the remainder of the cell body. So this is a nuclear pore, and the TDP-43 will move out into the cell body through that nuclear pore, and it can also return back into the nucleus through the nuclear pore. Now, at any given time, normally there's around 10% of the protein that is shuttling backwards and forwards between these two compartments. But for some reason, in motor neuron disease, TDP-43 builds up in the cell body. Now, it's not 10% of the protein that ends up in the cell body. It's not 20% or 30%. It's actually 100% of the protein ends up in that cell body. And what that means is that our instruction manual of our genetic information, our DNA, now no longer has TDP-43 there to interpret it. So our cells can no longer properly respond to our genetic information. And I hope that what you can see is that when TDP-43 is in the cell body, it actually forms these protein aggregates. Now today we're going to call these protein aggregates protein clusters, and I think it's quite easy to see from this demonstration that these protein clusters are now far too big to fit back through that nuclear pore and into the nucleus. So that's one of the ways in which we think TDP-43 causes motor neuron disease, the loss of the ability of our cell to be able to interpret our DNA instructions. But there is another way in which we think these TDP-43 clusters are important to motor neuron disease. And for that, we're going to turn on our presentation and show you our slides. So some of you might be familiar with this character. I think his name is Waldo.
Or Wally in Scotland.
Perhaps. So Where's Waldo or Where's Wally is a cartoon character that is put into a scene that is jam-packed with people. And the purpose of this cartoon is to find Waldo or Wally in this crowded scene. So I don't know, Melissa, can you find Wally?
It's a little bit challenging, but it's somewhere in the middle of the screen.
Oh, I see him in the middle with a backpack.
With the rucksack, yeah.
Yeah, so inside a cell, it's also kind of crowded. It's actually jam-packed full of protein. So what this picture is showing you is an artist's impression of what it looks like to be in that cell body. Each of those balls with different colors, that represents a different protein. Sometimes these proteins are in single components. Other times you'll see that there's protein complexes. That's where there's more than one color. These proteins, they've got to move around the cell body and they've got to find one another. And this is how they function or do their job in the cell. So as you can imagine, when you have a large TDP-43 cluster inside the cell body, it then makes it very difficult for all the other proteins in the cell to move around and to find one another. So now let's think about a brain cell or a nerve cell, as you may be familiar with. It has a cell body, it has a nucleus, that is the grey circle in the middle. It's surrounded by a cell body, and it has a very long projection called an axon. And at the end of the axon, there's finger-like terminals, and it's these terminals that then enable the information to be transmitted between brain cell to brain cell. It's how our arms move, it's how our legs walk, how we think. We really need these nerve cells to talk to one another. Now, when a cluster, a TDP-43 cluster, builds up in the cell body of a brain cell or a nerve cell, I hope you can see from this representation that it can make the information flow very difficult, if not impossible. So our lab is invested in understanding how TDP-43 becomes a cluster in the cell. And what Melissa is going to show you is the types of experiments that we do to be able to find out how these clusters are formed.
So I have a question for you. Have you ever overripened your bananas? You just forget them in your kitchen and then they become very ripe and you start getting these little insects, these little fruit flies that are quite annoying, and they're just everywhere in your kitchen. Well, these little flies, the fruit flies, we know them in the lab as Drosophila melanogaster, and that is our animal of study. Now, you might be asking why we're choosing to use a fruit fly. We have chosen it because for many years, scientists have deeply studied Drosophila. We know their genes, we know how they act, we know many things about them. And they are very similar to humans in their instruction manual or their DNA, how we were comparing it before. They're very similar to one another, especially disease genes. Now, what we care about in the lab is that the Drosophila or the fruit fly has a brain. It has about 100,000 different neurons, which is impressive. And we also care that it has a spinal cord. And that makes it good for being our animal of study in a motor neuron disease. So what we do in the lab is that we put these TDP-43 clusters inside of the fruit fly. So when we put these clusters in the eye of the fruit fly, we're able to see that there's a loss in the tissue structure. And we can also put these clusters in the nervous system of the fruit fly, and we can do experiments like this one. So what you can see here in the video is that the fruit flies have this reflex that whenever you tap the vial on the bottom, they will quickly try to climb up. That's just a reflex they have. So what we do in the lab is when we put these clusters in the central nervous system of the flies, they will struggle climbing up, or they will not do it as fast. About 80% of the flies will not climb. And because we are scientists, we like to make graphs about it. So here in the blue bar, we can see the healthy flies, and we can see in the black bar the flies that have those TDP-43 clusters. and less than 10%, about 5%, can climb the vial after 28 days. We can also assess how long these flies live. So here in the blue lines, we can see the healthy flies compared to the flies that are in the black lines that have the TDP-43 clusters. And we can see that these flies die faster. So this has shown us that TDP-43 clusters give the fruit fly symptoms of motor neuron disease. But the big question is how? So taking you back to the eye, this is what a healthy fruit fly eye looks like. Now, this is what the eye of the fruit fly looks like when we put those TDP-43 clusters in the eye. Now, us in the lab, what we do is we play Wally or Waldo, and we breed these flies until we find that one fly where we see an improvement of the symptoms. Now, me as a PhD student in the lab, I am meant to find how these symptoms are being improved. So there's one more thing that I want to show you, and that is a day in the life of a PhD student. It's a small video I did of what it's like to be a scientist investigating motor neuron disease.
Hi, I'm Melissa, and this is a day in the life of a PhD student investigating Alzheimer's and ALS. I wake up in the morning, I get ready, and then I head towards the Life Sciences building of University of Dundee. Here's where I'm doing my PhD. So first thing in the morning is to collect some fly virgins. My animal model is the Drosophila melanogaster. It's a fruit fly. Here's how we differentiate males from females from virgins. And it's important to collect virgins for the crosses just to make sure that we're monitoring the genes that we're crossing. Then I put them at 25 degrees where they will stay the next 10 days. Then some other flies that I collected that are still asleep, I take them to the fume hood where I use some chemicals to sleep them further so we can take some images of the eye. Here's a healthy eye compared to a diseased eye. The genes that we're expressing, the mutations that we see in Alzheimer's and ALS, we're expressing them in the eye of the Drosophila, just so we can see neurodegeneration and we can quantify it. There, I'm in the office organizing and doing some stats. Then I go for a little break for lunch. Afterwards, I come back to the fly room because I have another time point in the afternoon for another experiment. This is a little bit more complicated and a little bit longer because we are turning on genes only on the adult flies and they need to be monitored daily. So that's what I'm doing, labeling, making sure that they're doing fine, quantifying.
One eternity later.
Then afterwards, I'm done and I go back to the office just to do my last experiment of the day. It's a Western blot. This is an experiment that takes many days, but here I'm on the last stages of it. Basically, we have a membrane that has some protein extract, and we incubate it with some antibodies that will identify the proteins that we're looking for. Then we add this reagent that's called ECL, and we add it to the membrane. That will react with the antibodies that we have previously incubated the membrane with. That will be identified by this fancy machine.
A few moments later.
And then we can see the proteins of interest. This is a very interesting Western blot, actually. Then I pack everything and I get ready to leave work. And I go to Crieff to do some horse riding. That work-life balance is very important. So doing a sport is always recommended. Then I go back to Dundee at night. And that's basically it for the day. Then I'm ready to do science the next day. Thank you so much for listening.
Thank you, Melissa. That was a really nice video and excellent presentation. And we just want to show you some of the other members in our lab. James Gray, Charlotte Hurst, and the last picture there is Melissa. And we've had opportunity to work with many young undergraduates and rotating postgraduates, some of whom have been inspired to continue researching into motor neuron disease and related disorders. And we also would like to thank our funders, who you can see on this slide, and who really enable our research to take place. Thank you.
Thank you.